I'm starting now.
So we can start now.
Okay. Am I audible?
Yes, sir, you are.
Okay. So good evening, everybody. And this is the last of the lectures in the series from Mechanobiology, Principles and Application. So what we would see today is the application mainly. The experiments we have already covered the last time. So the application or the applications are mainly to study the response of the cells in the presence of a stiff matrix, or by matrix I mean the extracellular matrix, or a soft matrix. Also, we shall see how we can manipulate the cells, the ex vivo, and how we can measure the elasticity of the cells by micropipette aspiration. And at the very end, we would see the applications in the tissue engineering. Okay, so let us first see the effect of the matrix stiffness. So when you have the cells, when you culture the cells on the cover slips, you just put the cells on the cover slips, right? And the cells get attached to the cover slips. If you want to visualize the cover slips later on by microscopy, you fix the cells and then you proceed as you design the experiment. But if you want to design a matrix based on which you assume that the stiffness of the cell or the elasticity of the cell would vary, you can very well do it by using different substances. The most common one, very routinely used and have been used for a long time, is acrylamide bisacrylamide, okay, in specific ratio. So you are all familiar with acrylamide and bisacrylamide. You are all casting gels, the polyacrylamide gels. It's the same acrylamide. And these acrylamide you actually mix in the different concentrations. You polymerize it on the cover slip, and what you get are the types of matrices of the different stiffnesses. And these are the images of from the scanning electron micrograph, which shows that the polyacrylamide gels have different polymeric network as a function of the crosslinker concentration. So more you increase the concentration of the bisacrylamide crosslinker, you get a more and more compact network. Okay, so more and more compact network means more and more stiffer matrix. Okay, so then let's see how that affects the behavior of the cells. So when we make such matrix, we add increasing amount of this bisacrylamide crosslinker, and like here it's 0.5, and then gradually you go up to 30 microliters, and what you see is the stiffness increases along this vertical direction. So first you make the acrylamide extracellular matrix, acrylamide, polyacrylamide hydrogel, which is the extracellular matrix, on the cover slip or on the petri dish on which you are working, on which you are going to put the cells, and then you put your cells on top of that, and then you see what are the responses. So here, this paper is a fantastic paper. It talks about the podocytes. Now, where are the podocytes present? They are present in the glomerulus. They are the basic filtration unit of the kidneys. And the podocytes, as the name suggests, if you see the podocytes, you will see that there are the feet-like projections from the basal level of the cell, and they actually interdigitate the cells interdigitate amongst themselves, and it is through these interdigitate arrangement that the filtration occurs across the glomerulus. So the podocytes are plated on the matrices of increasing stiffness, as you can see in the panel A, and you can see the cell becomes increasingly stretched. These are the actin staining. You will see in the bottom panel that these are the nuclei. The green ones are the actin, and this is the protein called podocin from the podocytes, of course, which is specific for podocytes. So they become, the expression of the podocyte of the podocin becomes less and less on being stretched on stiffer matrix, which means that you will see that in many of the renal diseases, the, the elasticity of the cells, of course, the basement membrane, when it is below 2.5 kilopascal, it leads to disease scenarios, the various types of the renal failures. So people are interested in studying the microenvironment of the podocytes and what happens if you correct that environment, if you increase the stiffness of the extracellular matrix, do that or does that correct the organization of the podocyte, and does that reverse the renal diseases? Okay, so this is the basis of the mechanobiological studies on the podocytes, and you see that these podocytes become increasingly larger, increasingly spread on the surface as the stiffness of the surface increases. Okay, so you can read the details about this in the references, which are given at the end of the slides. So here, this is a fantastic paper. You can read this one for the details. And let's move on to the next slide. So this is this AFM image, and this is actually characterization of the stiffness of the cell or the elasticity of the cell. And you see that with the substrate elasticity, with the increase in the substrate elasticity, the cellular elasticity also maps in a similar fashion. And as you can see that this little key here, this index is like this, the white one starting from the brown one. It indicates that the surfaces of more of more elasticity. It's more stiff. Okay, and similar is the index here. So which means that in a nutshell, if you increase or if you modulate the surface elasticity, that information is transferred, is transmitted into the cell, which in turn modulates its elasticity in a manner to respond to that environment. Okay, and we shall see how. So here is another example of inducing stress on the cell. First one, as you saw, is by increasing the matrix, increasing the stiffer, which is one way of putting stress on the cell, and the cell responds to it by becoming more elastic or plastic, depending on what sort of stress you are giving. Here is another one, which is micropatterning. You may have heard of a technique called lithography. So there are various modes of lithography. You can actually print anything on PDMS, polydimethylsiloxane. It is actually it's an polymer of silicon, where you can actually make the mask and you can imprint the mask on the silicone, which leaves its impression, as you can see here. And there can be a photo patterning, there can be laser patterning, and based on that, there can be soft lithography and other different methods. So let's not worry about the method of lithography for the time being, but lithography can pretty much etch all the patterns, like it can etch the face of Einstein. Also, it can etch the different surface topologies on your PDMS membrane, on which if you grow the cells, which actually induces the differential stress, to the cells. Okay, so you will see that these structures will project out of the uh, of the basement membrane, and in that way, if you put a cell on top of a structure like this, so the cell actually falls. Okay, it drops from all sides of this structure, and if there is a hollow well into which you put a cell or you micro confine the cell, then also it's another sort of stress on the cell to which the cell adapts differently. All right. So these are the techniques which are being routinely used. It's not very new. It's almost a decade old, and people are using it, including our lab, where we are using the lithography techniques and we are using these micro patterns to confine the cells and see what are the effects of the actin, what are the effects of these the focal adhesion complexes or the branching of the actin. You know, the green ones are the stress fibers, and these are the various shapes what you can imagine. So pretty much you can imagine any shape and you can make your mask, and then you can put your mask on the PDMS, can cure the PDMS, and then you can put it on the cover slip and you can grow the cells on top of that. Of course, you need to treat it with fibronectin or collagen before that, so that the cell grows only in the areas which are coated with fibronectin and collagen, and or both. Okay, some people also use laminin, so it's your choice. So the cells will grow not directly on the PDMS, but PDMS treated with these proteins, okay, which you have already seen. These are associated with the extracellular matrix. All right, and this is the summary, which this is a very nice review, and you can see that all sorts of shape, like different squares, a pentagon, then a star, and this oblate spheroid and stuffs like that. So what happens to the cell fate? So you actually make these little whales in the PDMS membrane, and you put the cells there, which means the cells will be growing in the whale. So this is a representation of what is there. So if this is a square whale and you confine the cells there, so the it will take it will take the structure, and then what are the different biochemical cues which are elicited as a result of confinement of the cells there. These are all summarized in this paper. The references are all given. If you're interested, you can go to these references. There's absolutely fascinating studies on this. Now, this is a study from Shiva Shankar's group. It's absolutely fascinating. So there they made actually the different shapes and sizes, like the triangles and the circles, squares, and these rectangles, and they confined the cells on in these structures and this topography, I would say. And what they did the gene expression analysis. So as a result of confinement, you see across this axis, these are the different shapes, and these are the areas of this thing. So all these things have different aspect ratio. I'm not going into all the details, but you can pretty much have a bigger circle, a smaller circle, a bigger square, a smaller square, so on and so forth. And you can see, if you see the clustering of the genes after you do the gene expression profiling, and if you cluster the similar genes which are differentially expressed, you will see that for this circular size, these are the genes, that is the genes associated with these functions are upregulated. Similarly, there are the downregulated, upregulated, downregulated for various structures of various aspect ratio. Which means, which tells you that this stress which is imparted to the cell is manifested in the form of differential gene expression. See, here the main point is mechanobiology, as I'm emphasizing from the very beginning, is you impart some force, you see the response from the force. And the response from here is the gene expression. Okay, this is one of the expressions. The cell can change its shape, the cell can change the shape of the organ, of the organelles, which means also the nucleus, which means also the chromosome territories, which I think explained in the very first class. And as a result, you can see the differential gene expression. And there are also similar studies, more of these studies on the different sizes and the aspect ratio and shapes. And these are the genes. Okay, so this is from, as you can read the graph very well, this is like self-explanatory, very simple, minimum expression to highest expression. Okay, so you can understand which one are down-regulated, which one are up-regulated, and you can see these various genes which are upregulated. When you compare between, let's say here, a small triangle versus a bigger triangle, a small circle versus a bigger rectangle, a triangle versus circle. So you can imagine all sorts of things according to your need. You can design the cell type, you can design the shape, you can design the topography of the extracellular matrix, confine the cells there, and see the response of the cells in terms of the gene expression, in terms of changes in morphology, which is also another manifestation of the gene expression, in terms of the mobility or the dynamics of the cell on the extracellular matrix. So there are various methods to look into it. Again, some more examples of this. This is the staining of the actin, and you can, just quantify all these different things by confinement experiments. So these are basically the confinement experiments based on the shapes of the wells. Now, here is one modification of these experiments, also from Shiva Shankar's group. It's a fantastic paper in MBOK, and it states that they first patterned these rectangles on the fibronectin coated micropatterned dish. So as I mentioned, fibronectin is just here in these red things. So the cells will grow only there. The rest is PDMS. The cells cannot grow under naked PDMS. Okay, so they have to be treated. So the wells are there, which are treated with the fibronectin, and cells are growing. So you make the cells which are of this shape to become rectangular. So you are confining the shape, the cells in the shape of the rectangle. And on top of that, you put weights. Just the simple weight. You must have done the practicals in your 12th or your 10th when you put the different 200 grams, 15 grams on the Sartorius balance, when you learn how to weigh things. And so one of these weights, you take the different weights. So what's the principle? This weight, if it's like mass 200, mass times g, so like it's like 200 grams, so mass times g, that is the force exerted on the cells. Okay, so it is when we say the stress, it's actually a force per unit area, right? So you can very well calculate the stress imparted on the cells, and then you see what are the effects of that. Fascinating effects. Now, this is the DNA stained with the DAPI, and when you increase the load, when you put the load in the first place, so what would be the control? The control would be no load, right? Just patterned, no load. Patterned, load. Okay, so this will be the control. When you think of an experiment, you should always think of the control which would give you which would enable you to interpret the result. So if the result here you want to interpret is the effect of the load on the cells in the pattern dish, then the control will be the pattern with no load. But if you want to study both the parameters, that is cells confined in the micropattern as well as the load, then your control should be cells on the matrix which are not patterned and no load. Okay, so then there will be the two variables. Okay, and so you can vary the variables as long as you want to make your experiment much and more complicated, and then you can do principal component analysis to boil things down to a lower dimension. Not going there, here just what we see here is when we put the load, we see that the DNA is much more content. So it's actually basically we are doing the quantification of the dapple staining, which condenses the chromosomes. It becomes much more heterochromatic. So compared to this one, the control, and when we put the load, it becomes clearly visible that the heterochromatin content increases. Similar thing we can also see they proved by these epigenetic markers, which are actually the repression markers, H3K9, H3K27, the trimethyl. So which means that these markers, if upregulated, so more of a heterochromatin content, the gene expression is shut down. Okay, so the load has an effect on the organization of the change in the spatiotemporal, let's say space and time, spatiotemporal organization of the chromosome, and also on the epigenetic landscape. Okay, now you imagine, you pause a moment, you imagine the stress or the shear stress. Let's say, for example, the endothelial cells on the uh, the blood vessels, the lining of the vessels. So it is actually experiencing the constant flow of the blood. Okay, it's being pumped to the heart at a high pressure. So you see there is a shearing force on the cells. What might be the effect of these shearing forces so that the cells keep on maintaining their function? Okay, what happens in the cases of the cancers? Okay, so we will see actually that the different types of cancers, they have a differential interaction with the extracellular matrix, and there are studies, including our group, that we are seeing that if you modulate the stiffness of the extracellular matrix, you can actually control the metastasis of the cancer cells, the migration of the cells from one part to another. And these are some more examples of the staining of the markers of the nucleus. But just to cut a long story short, stress on the cell in the form of the extracellular matrix confinement, or putting a load on the cell, putting a shear force on the cell, induces gene expression changes, and that depends on what sort of shearing stress you subject the cells to. Okay. Sorry. Now we gradually increase the level of the complication. So you can now imagine that you can make, not imagine, you can actually make whatever you can think the micropatterns on the PDMS membrane you can make. As the name suggests, they are pillars, right? They look like pillars. So you can fabricate these pillars on the PDMS membrane. So these pillars are also PDMS. You can make the pillars of different stiffness such that if the cell is plated on the pillar, the cell would exert a force on the pillar. So the pillar can bend like this, okay? And you can actually measure the oscillatory frequency, oscillatory motion of these pillars. This is one type of measurement what you can do. You can have some microsphere bead embedded between the pillars, and you can do a traction force microscopy. Or in another scenario, if the pillar is stiff, that is, the pillar is not deformed when you put the cells on top of that. So if the pillar is soft, when you put the cell on top of that, the pillar buckles, right? But if the pillar is stiff, it actually pushes the cell, which means it exerts a force on the cell by which the force has to respond, or the force will change its morphology, or the cell will change its morphology, the cell will change its function based on the gene expression. So here are the different types of the pillars. Again, the fabrication is by lithography. So these pillars are called micropillars because they are the micron the dimension. Similarly, you can find structures called the nanopillars, the nanoforests, the different kinds of the the lithography, different kinds of topography, what you can think of, and which you can fabricate by the lithographic techniques, and then you can study the differential force on the cell. So here are these mesenchymal stem cells within the micropillars for 3 weeks. And the pillars have the different spacing, which means if you go back to the previous slide, there is a distance between the pillars. Okay, so you can vary the distance between the pillars. So they can be 5 micron, 10 micron, 20 micron, and the cells are stained with actin and the nucleus. And you see the differential behavior of the cell, 5 micron, right? So the pillars are very close to each other. 10 micron, the pillars be apart, 20 microns they're apart. And you see that has a substantial effect on the morphology of the cell, okay, and the cell number. So the pillar spacing has also significant effect on the cell number. Okay, so if the pillars are widely spaced, more cells can grow in between the space, and which is contrary to the case where you have less space between the pillars. All right, so then you see from these the confocal micrographs, which is absolutely beautiful. You see this little hole here. So this represents the projection of the pillar. That is, the cells are growing encompassing the pillar. So keeping the pillar here, the cells are growing like this. Okay, so the cells are growing in and around the pillar. It is encircling the pillar. And that's why there is this little hole here, and the cell can assume all the different shapes. You see the two cells are fused here with the two pillars in between, and these are stained with the different types of stains of vaccillin for the focal adhesion, nucleus, actin, and so on and so forth. So the cell structure changes. Okay, tubule. So the microtubules, as you know, they change. Actin, microfilament, they change. Nucleus, they change. And based on the topography of the surface. As I was mentioning, you can also measure the dynamics of the cell. You can actually take you can take time-lapse imaging of the cell between these pillars, and you can then we can actually plot the trajectory of these things, and you see that of course, this is understandable that the more space you have, so this is a pillar with more spacing. Let's say the 20 micron is a surface topography with more spacing between the pillars, which means the cells can move around in between the pillars with much more degree of freedom compared to the scenario where it is 5 micron, the spacing between them. Okay? So and you can quantify all these things. You can collect the number of events from the different types of cells. So here we wind up this topic of engineering the different topography and the different stiffness of the extracellular matrix, which would indirectly exert an effect on the cell. And now we come to a technique called micropipette aspiration. This is the microscope we use. I typically have this microscope in my laboratory. This is a micropipette aspirator microscope, and here you can see the two arms of the manipulator. So this is the micro manipulator, and at the mouth of the manipulators, you can attach the fine capillaries, and there are two such things. You can use the two manipulators together, and with this joystick you can actually move it, and you can suck the cell inside the microcapillary, which is attached at the tip of this manipulator, by applying a negative pressure. How? I will show in a minute. So by this, you are no longer modifying the matrix. So this is another way of looking at things. This is perturbing the cell or directly manipulating the cell in terms of the force. So you are actually wrenching the cell, you are twisting the cell, you are physically distorting the cell without the involvement of the matrix. So matrix indirectly transmits the force to the cell, which is deformed. But here you are deforming the cell and seeing a response from the cell. We'll see the mathematics behind it, the principle behind it. So first let us see what is the principle. So this is the cell and this is the live cell. So this is the microscope objective, CCD camera. You can see it in the computer and you can manipulate looking at the computer with your two hands working on the joystick, which actually manipulates the manipulator. So with one manipulator, you catch hold of the cell, you make the cell, you fix the cell at one point, and with another manipulator, with the microcapillary, you suck one portion of the cell into the capillary. Okay? Now, how do you suck things? You actually apply a pressure, and by the pressure you apply, there is a pressure gauge actually, which is adjustable. So as you know that the delta P, or the change in pressure, is rho G delta H. So the height at which this little trough is placed is filled with the fluid. It actually manipulates the pressure by which it sucks your cell inside the micropipette. Okay, it needs fine precision, it needs a lot of practice, but it is pretty doable. A lot of labs use it. My lab also has this microscope, and we do this thing. And so this needs a lot of manipulation. And there are several things which you would notice here. There are the things called LP, that is the length sucked inside this capillary. RC, that is the radius of this, the cell, which is sucked in. And then you can apply the different pressure, the different times. You can modify the ramp of the pressure, and you can see all sorts of behavior of the cell in terms of being sucked inside the capillary. But you should be careful that the force should not be such that the cell ruptures. Well, of course, many cells will rupture in the process, but then you collect this information from many different cells, you have statistics, and then you actually present this thing. So this is the little formula what we use here. The delta P is rho GH and is a correction factor 1 minus U by UF, where U is the velocity of the aspirated material and UF is the velocity if it would have flown freely, which means the cell, if it would have flown freely means there is an element of viscoelasticity. As I mentioned, that there is some viscosity, some element of viscosity also there in the cell, which means that if that viscosity allows the cell to flow, what would be the velocity of that flow? That is the, without any aspiration, that is UF, and 1 minus U by UF is the correction factor, which gives you the delta P is equal to rho GH times this one. Okay? And you can pretty much quantitate, and these are the various applications of the micropipette aspiration based technique. It can be micro indenter. You can actually use to indent the cell, poke against the cell. You can micropipette aspiration, the one which I was discussing. And you can use the cell pushing and the pulling, cell detachment technique, the dual pipette aspiration. The microaspiration device was used to map the surface tension of the cells. So, it has many different applications. So this little technique has been used for almost a decade or even more than a decade, and it yields wonderful result with respect to the surface tension, with respect to the overall stiffness of the cell. And then we see how it can do it. So these are the references if anyone is interested to go deeper into that. These are the models which are involved, and these references are there from this paper. So if you go to these references, this gives you the detailed model of the material which are being aspirated, what is the change in the geometry, different types of solid, and different types of fluid, transition between fluid and solid, and what are the assumptions, what are the models there, and the mathematics behind it. So, but to put in a nutshell, what basically happens is what we measure is the differential pressure and the aspirated length that we can measure. I will show you in the next slide how you can measure that precisely. RP is the pipette radius, that is the micropipette radius. You know you have taken a micropipette of a specific thickness, so you take the inner radius, then there is the 5P depends on the thickness of the pipette wall, and the slope of the delta P by E is given by 2 pi by 3 5, that is roughly equal to 1. And this model may be extended to a linear incompressible viscoelastic material where this LP can be taken as a function of time, and elasticity is replaced or can be replaced by a creep function. Do you remember the term creep function, which I had discussed earlier, I think in my last lecture, that this is constant stress. That is the behavior of the cell, the viscoelastic material, in response to a constant stress. So it gradually changes with time. Okay, so this is the creep function, and this equation takes this form. So we can measure this LP and RP. How? Let's see. So this is the image of this micropipette. This micropipette, you are bringing it close to the cell. You puncture through the plasma membrane. You are interested in the nucleus. Make sure you don't rupture the nucleus. This is the nucleus when it is not being sucked in, and this is the nucleus which is being aspirated. Okay, you see the nucleus enters into this micropipette, and you calculate this length, the distribution of the actin. The force is in this direction. You can make all these different calculations from the GFP laminae and the nuclear pore complex stained with the different colors, with the different fluorophores, and this is exactly what happens. So as I mentioned the term, that is the creep. So with the creep, you are applying the constant pressure. You see more and more of the cell is being aspirated inside the micropillars. Okay? So this is the creep function. This is the visualization of the creep function. Okay, and from here you know that after a certain time what is the LP, and you actually fit the LPM, sorry, according to the various functions. I'm not going into there, but these functions are available. The literature is wide and very deep on this subject, so you can, anyone interested in this micropipette aspiration can find many literature, many different applications, calculations, and you can easily calculate LP, RC, and from there you can actually calculate also the stiffness of the cell or the creep of the cell. Okay, fine. Now again we come to another sort of microfabrication. We make, or we can make, the very complicated structures like the one it is shown by Ben Fabri's group, and this is a very complicated one, a very complicated design. This represents the various capillaries, microconstriction capillaries, which are printed on the PDMS membrane, and that membrane is actually put on top of the cover slip. That is, it makes a microfluidic chamber. Okay, and these cells, you make the cells flow through it. Okay, so you actually put a pump, a perfusion pump. You actually flow the cells and the medium through this perfusion pump. You attach one to the inlet and one tubing to the outlet, and you flow the buffer. The cells will actually pass from this end to this end through these microconstrictions. So the junctions actually represent these microconstrictions, and this is actually the profile of the cell And if you follow the profile of the cell, it gives you very many information about the structure of or the deformation or the change in morphology or the change in the elasticity, which actually leads to a change in morphology. So this is also another microfluidics device from Lautenschläger's lab, and they call this system microfluidic microcirculation mimetic. So here they have a question. So you design your microfluidic chamber for the circulation according to your need. So the basic thing you have to keep in mind is the neck of the microconstriction should be substantially smaller than the nucleus, which is a very hard organelle of the cell, so that the nucleus has to squeeze through it, and that will be a test of resilience of the nucleus when it passes through the microconstriction. Okay, so you should know in the first place what type of cell you are choosing. The design of the microconstriction, microcirculation, microfluidic system depends on what type of cell. Suppose you take a rectangular cell, big cells, you need to have a design of the microconstriction not too small such that the cells while passing through it, they rupture, but just small enough so that they can wiggle through it, right? Also, if the cell is small, the diameter of the cell is small, there are very different types of cells, then accordingly, you have to make the microconstriction diameter even smaller. Now, here the question is, what controls the dynamics of the cell when the cell crawls from one place to another, whether it's actin or it's myosin? So they have this cell HL60, and then they treat the cell with the blebistatin, which is actually an inhibitor for the myosin. And this is the advection time. Advection time is the time taken from here to here to traverse this entire length to come out of the outlet. Okay. And you see that with the blebistatin, it's almost similar, maybe a little bit more. The cell becomes a bit more stiffer than the untreated cell. But when you treat the cell with cytochalasin or latrinchulin, which are all inhibitors of actin polymerization, which means it depolymerizes the actin, which means the cell becomes much more fluidic. The scaffold provided by the actin is no longer there. And as a result, the cell becomes much more fluidic, much more plastic. Okay, so it can easily, it's like a chewing gum or something. It's like very wobbly. It passes through this microconstriction, at a very fast time, and you see the time coming down from, let's say, like 3 seconds to almost less than 2 seconds. Okay, so this is the median. So basically the question what you are asking, you treat the cell with whatever drug you want, and you want to see the treatment of the drug on the cytoskeleton. Can be microtubule, it can be actin, it can be intermediate filament. Design your experiment whether you need the microfluidic. For all the dynamics experiment, you need a flow, and for the flow you need a constriction. Okay, so if this is the model, then you have to choose based on the morphology of the cell, based on the treatment of the cell, and also the design of your experiment as such. So here is this paper from Jan Lemmerting's lab at Cornell. So they have manufactured this sort of very distinct microcapillaries, and the cell is, you can see this is the nucleus stained in red, and the cytoplasm, it's stained in the green. Don't worry about what stain they use. And you see the cell can make its way through it and see the different stages that with different times, the time-lapse imaging, the different frames, which tells you the different morphology the cell takes. Now it can happen, and it actually happens in the different mutations of lamin, that the nucleus is brittle, the nucleus becomes vulnerable to these forces, so that the nucleus breaks at the slightest assault, right? And this assault can come in this form of this, the confinement of the microconstriction and flow. So there are the two forces, the flowing of the cell, the buffer, the medium, and the medium flowing, the cells in the medium flowing through the microconstriction. So these imposes these forces on the cell, which actually gives you this sort of changes in morphology of the cell. You can quantify each of these. People have made models of this thing, which you can see here. They have made, they have simulated the models, and you see that in the case of the lamin A, you see this is the lamin A, which is there. Plus means both alleles are there, and this is a knockout. Okay, and you see that the volume is normalized volume is less than that of this thing, right? So the average transit time also, you can see that it is different in the case of lamin A minus and lamin plus plus, which means that the lamin A minus minus, when it is not there, when it is lamin A minus minus means lamin A null, knockout. When it's not there, it cannot exert the mechanical strength on the nucleus. It cannot impart the rigidity on the nucleus, so the nucleus flows much more easily. And these are the various modelings. So they have made the model, and these are, from these the simulations they have made, and they have shown that with and without lamin, what are the, how the contour of the cell changes, and they have quantified this thing. So as I mentioned to you, depends on the vision of your experiment, what you want to quantify. But these are the handles. The basics of this course is to provide you with an example of what are the different handles which are available to you, to you in various forms, and you can use it to answer the questions you ask. And then we have come to the very end of this course today, the applications in the tissue engineering. And I strongly recommend that you must read, or must go through the wonderful papers of Donald Ingber. Remember, Ingber proposed the tensegrity model, and he is the guy who is actually one of the pioneers of tissue engineering at Harvard University, and they have made all sorts of organs on the chip, which is based on the stretchability of the extracellular matrix and also on the physiological design. So they followed the physiological design as close as possible with these microfabricated devices, and they could manufacture, they could make cells which have the function like the physiological cells in the body. Okay, so these are the different chips, and this is actually the PDMS membrane, which can be stretched, and it comes in two layers. So they actually made an endothelium. This is a little filter, and endothelium actually is stuck upside down. So the cells are going upside down, and they are flowing the medium both through this chamber and this chamber, and this entire thing can be stretched. Okay, so in case of the lung, as you know, the lung alveoli, so they are constantly getting expanded, right? Because you are inhaling and exhaling the air, and it is these alveoli which are actually the sites for this gaseous exchange, and they have fabricated these cells, these epithelial cells, on the surface of the endothelium, which is separated by a semi-permeable membrane, which means that these endothelium provides the materials for the growth of the epithelial cells on top of them. Okay, so this is a physiological design. Also, in the context of the physiological design, within the tenets of the physiological design, they have applied the different stress, the different stretch. You can see the arrows which actually demarcate the direction of the stretch. They have very nice videos on Jove. You can go to the you can go to YouTube. You can go to his site at Harvard. And you can see all these different and wonderful the tissue engineering. I'll show you one more example. So here actually they have shown it from the side, which is that is the cross-sectional view. And so this is the endothelium, and the green one represents the epithelium. And they stain the endothelium and epithelium with the different markers. And you see that these are the different, these F, this, you see these the polygons, if you can see the polygons here. So you see that these are the different these are the different stretch inducing pattern on the membrane. It's actually a bit complicated. So if you read the papers, you will see the basic design and the basic need of this thing, and they give you the direction of the stretch. And they have also studied the infection of the lung epithelium by the E. coli, how the E. coli are lodged on the epithelium. That is exactly what happens if you have a lung infection, not by E. coli, but any other organism, that how or where the E. coli or the microorganism gets lodged. Okay, so this is a very, what they emphasize to show that on a chip, you can basically take out the entire organ. You can study the function of the entire organ. You can first synthesize that organ, and you can study the entire organ. And not only that, they have seen the response of the TNF, the neutrophils, which are actually secreted in response to this infection. So this is a wonderful model. This is just a glimpse of the tissue engineering approach, what they have here. And you can also design your own experiment. There are other papers also where they have designed the kidney cells and the different the heart cells. People have done the heart cells before of the rat, the Sprague Dolly rat. They actually made the heart cell beating heart in the petri dish. So that's actually pretty old. But now, based on this nanofabrication, you can actually make these human organs. You can make the function as human organs. You can follow the function of these organs. You can follow the biology, you can follow the biochemistry of these organs following an infection. See, now we have this pandemic raging globally, and now models like this, that is the lab on a chip, the organ on a chip, and if you have the models like lung, so that is the primary site for the infection of the COVID virus, right? So that will be a very pertinent model to study all those infection modalities, and also the treatment regimes only in the form of a chip, if you are interested to do so. And so here again, they study microengineered model of the pulmonary nanotoxicology. As I was saying, they study with the nanomaterials, and they see what is the effect of nanomaterials in the presence of the stretching. That is, the cells are constantly being stretched. So this is a busy slide. I don't have the time to read all this slide, but if you're interested, you can go to the details of this thing. It's a science paper, very elaborate, and you can see that you can measure, as I mentioned, all the biological functions of the cell ex vivo in the form of a tissue, in the form of an organ actually. Okay? And this is also the example of the microfluidic design, where you can actually culture the stem cells, and you can the CD34 plus progenitors, and you can make them a bone or something like that, whatever you like, based on the factor what the cell needs for its differentiation into that lineage. So basically, it is a PDMS based chip on which you can flow the different You can flow the different factors, which are the different protein factors, which are required for the formation of the different organ. And you can actually make the basically you can make the different organs based on this microfluidic chip. So this is also published in the Nature Biomedical Engineering, a very interesting paper. Those who are interested can go through it, and you can get a whole idea about how vast the field of mechanobiology is, how it is expanding, what are the innovations what have been made, and what are the scopes of innovation. So you have to think wild, you have to think big, think loud, and if you have a design, then you can design your own. You can design your own system. You can study your own system at your will. So I think we come to an end of this thing, and thank you very much for your continued attention for the three lectures. And I would be ready to have any questions if you have any at the moment. Let's see if you have any questions. So I guess there are no questions. Anurag?